Well, we stay in the spirit of the nPOD Pancreatic Organ Donor Consortium. In the next 15 minutes or so, I will tell you a couple of new things that have left many researchers, including myself, a little bit befuddled, but it also will tell you how much we can learn from this organ donor consortium. Then in the end, if there's time, I'll talk a little bit about therapeutic approaches. As a disclaimer, I will not discuss any Novo Nordisk drugs in specifics, of course. So when one looks at the pancreatic organ situation, there's some striking things. In the pre-diabetic phase, when you look at the changes that are in the pancreas that you would consider abnormal, they're few. There's also little inflammation. This is something that defies, in a sense, a dogma that we have learned over many years from animal models. And it makes us maybe gain a different outlook on the disease, and I'll illustrate this in the next slide. The other thing we have learned, and we will talk about these different players here, we have learned that the most frequent cell that we find is a CD8 cell that can also be a killer cell, in conjunction with an immune recognition molecule that is usually enables these type of cells to kill other cells. So probably a deleterious combination, and we will talk about this also a little bit more. And then there are several other things that go in the direction that Mark has already mentioned, that there's abnormalities in the pancreas, not only the islets, but the exocrine pancreas, and I will show you these too. So it's not a disease that is as simple as that there's an immune system defect that knocks off the beta cells. There's many other things that are maybe in the disequilibrium. If we stay with the first situation, maybe this famous curve that we have now redrawn many times in the last decades, probably. Maybe we have to redraw this curve, how beta cell destruction occurs again, or at least consider this. So here's some considerations. One is that the beta cell mass, and Mark already indicated this, might be smaller in type 1 diabetes because indeed the pancreas is smaller. And then in the pre-diabetic phase, when one has autoantibodies, and it's already a bona fide disease based on that, what is curious, you don't find that much inflammation in the pancreas. So we have to consider that it's a reiterative process that goes back and forth over some time, till at some point glucose tolerance becomes abnormal. What is even more curious, what we see at least in some of the nPOD cases as relatively new data, is that the beta cell surface area, we don't know about the function, but the surface area goes up just before the time of diagnosis. So that's also pretty curious because it means the beta cells have not completely lost for quite some time. And then just as we know around the time of diagnosis, the drop of the beta cell mass must be at least so much that is not sufficient to make enough insulin. Of course, we know this. If this is true, if it's reiterative, and it has this more precipitous drop of the time of diagnosis, and then it re-equilibrates with the honeymoon phase again there, then indeed the time to intervene, and Carla has spoken in quite some detail about this, is just what we call secondary prevention, before the diagnosis of the disease with multiple antibody positive situation and abnormal glucose tolerance, because there would be still more beta cell function and mass to preserve. So that's something that is something that comes out also out of the histopathology studies as a very interesting new finding in my mind. The other thing that you have to consider is that these destructive processes in the human body, if it occurs like this, they are processes that recur often in relapsing, remitting fashion, because things in our body are set with feedback loops, multiple feedback loops. The immune system is set like this. And you could consider this almost like a climate change, that this stays in equilibrium for a while, and one is okay. Then it really doesn't work anymore, and it re-equilibrates in another equilibrium. What comes with that is that actually beta cells that can be functional tend to exist for a long time, and Mark spoke about this after the diagnosis. So maybe some food for thought. Maybe this is how we should redraw the diagram. We can discuss it later on over coffee probably. Now coming to the other factors. The immune recognition molecule is called MHC class 1. When you see this here, in a sick islet, all the red staining is this upregulation of this MHC class 1. We have still no idea where that comes from, but it's a hallmark. You can take type 1, pancreas from a type 2 pancreas and tell them apart. You can also appreciate in green here the existing beta cells, and on the right-hand side in red, the CD8 cells, not so many of them, but certainly clear infiltration, and the cells that we think destroying the islets. Now, this MHC class 1 recognition molecule is existing up to many, many, many years, upregulated in certain areas. What is curious about it, it's lobular, and I think we had it brought up during discussion today before. See, here's a whole pancreas with destroyed beta cells. This is after onset of diabetes. Just one area where the islets and this recognition molecule are still present. Very curious, because you could ask, why is this not patchy across the whole organ? Why is this one lobe an area where these beta cells are still being preserved? So altogether, a quite interesting situation, and makes us ask many questions about this. We still don't know where this class 1 comes from. It could be induced by a virus infection that makes areas of the pancreas sick in a lobular fashion, maybe even a herpes virus that comes back and goes into latency and comes back and goes into latency. All of us have many of these herpes viruses, so that's a known phenomenon, and maybe there's a specific one that triggers these organ pathologies in a local area. Not impossible. If you think about the herpes virus in the lip, very specifically triggers pathology coming and going in a certain area. It could be other things that cause this. We really don't know. It could have to do with the innervation of the pancreas, and it could also have to do with certain immune mediators. We think that altogether, the combination of these CD8 immune maybe killer cells and this recognition molecule is not a good combination to have in the pancreas. I will tell you a little bit more about these immune recognition molecules and the cells. Now, relatively new data from the way we see them distributed in the pancreas is that we can attribute several self-antigens or auto-islet cell antigens being recognized by them, as listed here. It accounts for some of the cells we find in the islets, but we also find a fair amount of general virus-specific cells. And this is a staining that can identify a cell in the human pancreas and what the cell sees and recognizes as an immune cell directly, locally, where it's being stained and identified. So we don't know at this point whether the disease primarily is caused by the autoimmune cells, whether virus reactivity contributes to that. What we can know from animal models that just the presence of a few of these autoimmune cells can be bad. This you can prove in animal models. So in principle, it is possible that any of these specificities here of these cells is driving the disease. But we don't know that. We will not know this until we have a specific way of eliminating them and try to treat the disease this way. And there's ways underway actually working on both angles. One is on the virus angle to reduce certain viruses like enteroviruses that are thought to cause the disease. There are groups in Finland working on this. And the other ones are antigenic therapies that are trying to specifically eliminate some of these cells that we think could destroy the pancreatic islets. There are several other things I wanted to mention to you because they're new. I didn't expect them. They're curious type of things we find in the pancreas. We find, for example, that the exocrine pancreas, so the whole area where the beta cells not are, is usually also inflamed to a certain degree in type 1 diabetes. That's something that we have seen in all of these nPOD cases, and it's not related to the way the pancreases were acquired or harvested, and it's not related to any other thing that we can attribute this to except for the type 1 diabetes diagnosis. We can also find, and these are interesting things, we talked before today about the differential diagnosis between type 1 and type 2 diabetes. We can find some autoreactive cells in type 2 diabetes. Jerry Palmer and his team have worked on that as well. We can, again, in the blood only find these autoreactive CD8 cells in type 1 diabetes, indicating that there might be a specific destructive cells. We have looked at immune mediators, such as cytokines, in this case interleukin-1, and it has a curious distribution in terms of its highest levels. I'll show you that, mostly in type 1 diabetes with a high BMI, indicating that it might not be so much a sign of diabetes, but maybe of obesity. And the lobular affectation of the pancreas we already discussed. So I show you some data about this. We quantitate now in these organ samples with a blinded scan system the cells that we find either in the islets or in the exocrine pancreas. And in general, when you stain here for the CD8 cells, this is how it looks. You also appreciate in this case, for example, that the cells that are in the islets are not necessarily always more than the cells that are outside the islet. So the disease presents itself often, especially in the pre-diabetic phase also, that there's some infiltration in the exocrine pancreas, maybe implicating the whole organ in the cause and the development of the disease altogether. And that gives us new ways how to look at the disease and maybe also new ways how to prevent the disease. Mark always says this, the more we study nPOD, the more ideas we will get about new therapeutic approaches, and I strongly believe in this. So here you can see in the antibody positive state in the type 1 diabetes, also in type 2 diabetes, when you quantitate CD8 cells, but also other infiltrating cells in the exocrine pancreas, they are clearly elevated. When you look at the overall spread, there's a fair amount of a spread. What we need to find out now is why in these cases where they're particularly high, this may be links to certain features of the disease, maybe a more deleterious course of the disease, maybe certain other metabolic factors that could be linked to this, and we don't know this at this point. The bottom line is that the exocrine pancreas is clearly involved in the disease. And you can also appreciate here, when you look at what is driving this, when you look at cells in the islets, they're clearly driven by insulin. Many of these will only be seen in the islets when they are still insulin-producing beta cells. The cells that are in the exocrine pancreas, in type 1 diabetes, insulin-positive islets, insulin-negative cases, you find them any which way. So that indicates that the islet inflammation is something specific, also driven by insulin-positive beta cells, but that there's other factors that occur in a type 1 diabetic pancreas that might contribute to disease that are not driven by the beta cells, but by other factors. Whether these are virus infections, whether these are co-phenomena that are not directly related to the type 1 diabetes, we don't know at this point. So just showing you one slide here of the reactivities that's now looking in the blood. We have been looking for cells that recognize the own proteins of the beta cells or autoantigens in the blood. And you can see here, and they make different type of immune mediators. You can see here that in type 1 diabetes, as well as in the type 2 diabetes, in many cases, you can find these cells specific for islet antigens. That indicates that maybe in type 2 diabetes, also a certain amount of beta cell stress can lead to interest of the immune system to the beta cells and to autoreactivity. And Jerry Palmer's data have indicated so far that he has published that in those cases with type 2 diabetes, where there is an interest of the immune system for the islet antigens, the C-peptide or the beta cell function is lost more rapidly. If that's the case, we can, in a way, redraw our understanding of the diseases. Maybe we have better down the road to differentiate between different type of syndromes. One is an autoimmunity syndrome that's typical type 1, where beta cells are recognized, mainly CD8 cells maybe. Then there's autoimmunity that can be linked to beta cell stress, also CD4 cells. You can find it in type 1 and type 2. In both cases, if that's occurred, it's not good, and it would invite an immune therapy to lower these type of reactivities. And then you're going to have many other factors that are of metabolic nature that contribute to the disease, and they can also occur at the same time. It could be a high BMI, it could be insulin resistance, it could be these type 2-like syndromes. And Bill was saying it very nice this morning in the early talk, that these type of things can happen at the same time. There's no rule that only type 2 or type 1 things can happen at different times. They can occur together, and I think that's what we clinically see also more and more. And it will also ask for specific drug treatments for these type of syndromes, rather than just saying this is type 2, this is type 1. I'm also not so sure after seeing all this pathology that LADA is really pathologically a completely different disease. It might just be like a type 1 diabetes that has occurred much slower and then is overlaid with type 2 types of symptoms. So when we look at cytokines, there's also some interesting lessons. It's something we have just started. with the organs. These are immune mediators, how cells talk to each other. This is one of them. There's about a gazillion of them. So we will work our way through the most interesting candidates. This is one that was used in clinical trials in the form that people tried to block this in type 1 and type 2 diabetes. It is therefore interesting to see how these cytokines might be distributed in the pancreas. And we look at this on the production level for the genes for the cytokine, and we also look at this on the protein level for the cytokines. And what you can see here is that the elevation of the cytokine is not following a very distinct pattern that it would only be in type 1 diabetes or in type 2 diabetes. Actually, curiously enough, if anything, it's related to a higher body mass index and the upregulation of the cytokines, but uniformly, it shows a very, very, very mixed pattern in terms of inflammatory cytokines. And it might also therefore explain why blocking the cytokine has not been that specific. I think this is, in our opinion, one of the first studies where we're trying really systematically to go through and find the levels of a cytokine. You can imagine if you look at fewer cases or fewer islets, you have very easy time to pick your favorite islets and say it's either high or it's low or it's middle, but it's a lot of variation, as you can appreciate in many of these human studies. And in this case, I might explain why overall, in general, it didn't have much of an effect as a blockade in type 1 and also not that much in type 2 diabetes. Maybe at the end, just a few words about treatments. I don't know how I do with time. We're running over. Okay, 2 minutes I can do, if the slides advance. So let's spend just these last 2 minutes why it has been so difficult to treat about type 1 diabetes and what we could maybe do about it. If it advances, let's see how that goes. So there's 2 points I want to make. One is that the art of doing this is going to be very complicated. And it's very nice that now, as you can appreciate from the meeting, many people work together of doing this. This is not going to be a one laboratory or one company type of solution that we can offer here. But certainly, one issue is that striking the right balance between benefits and side effects when you modulate the immune system is going to be and remain a challenge because of a permanent therapy that might be needed to keep the destructive immune system at bay and the good other solutions to at least control the disease so that the willingness, understandably, to have long-term immune suppression wouldn't be so high. It wouldn't be also from my side. So for that reason, I think one solution would be that we will need combination therapies. Combination therapies have been very successfully employed in the oncology area with the main purpose to spread side effects and to have more impact where it matters, and sometimes also resulting in lower dosing of drugs that are needed, but they can be given at a lower dose if it's a combination therapy. We would suggest that you could split them into maintenance components. There could be antigenic therapies that induce tolerance together with GLP-1s, may be easier to give for a longer time. And induction therapies that could be anti-inflammatory or against the T cells and B cells that are found in the islet. So at the end, we might need all 4 components or a smart combination of these type of things. The second thing I think we will need is some smart type of biomarker to group patients when they're either in a recent onset trial or in a secondary prevention trial, in those who progress faster versus those who progress slower. It has to be done in a very targeted manner, in our opinion. In mice, at least, for whatever they are good, you can see that mice who have more of these CD8 cells progress faster. The ones with the lower level of the CD8 cells progress slower. If we have something like this, simple biomarker, then we could maybe have trials in a more concentrated fashion with a faster endpoint depending on what type of group of patients we can pick. So these are 2 things that are dear to our heart and that we are trying to implement. I leave you, I think, at the end with a movie altogether. This advanced and maybe. Anyways, and while they might be trying to get the movie going, I want to thank the people. This histopathological work mostly done in La Jolla Institute down in San Diego, independent from Novo Nordisk, and the interventions and strategies are, of course, done here at our center in Seattle. This movie, when it runs, it shows you the search and destroy mission in the mouse in life of CD8 cells, how they can go about find islets in the lobe of the pancreas. You can see there's a 30-minute time lapse. You can see how much these little guys can woozle around. It's like ants on the kitchen floor. It's a very dynamic system. It's also very interesting, my last word, is that they come about, and it's all random walk while they're in the exocrine pancreas. They don't know anything about the islets. Only when they're hit by random, these islets, they slow down and find their targets. So it's very interesting how the immune system goes about spreading and finding things. There's going to be this random deployment, and then by this random notion, they increase the likelihood of specific hits. And this is, I think, also how it occurs in type 1 diabetes.
Thank you.
Wasn't too long.